So now we will see some experiment to prove that DNA is a genetic material. We are talking about that the DNA is a genetic material. Genetic material means passed from generation to generation, right? So to prove that, there are some experiments shown by some scientists. Okay, we need to discuss 3 scientists experiment to prove that DNA is a genetic material. The first experiment was done by Griffith. Okay, so the experiment is known as Griffith experiment on transformation. So the name of the experiment is known as transformation. Griffith transformation. Griffith experiment on transformation. This was the first experiment performed by scientists to see whether the DNA is a genetic material or not. Genetic material means that passed from one body to another body. Okay, that is generation to generation. So in his experiment he used 2 things: a living mice, okay, a living mice and a bacteria called Streptococcus pneumonia, NIAE, Streptococcus pneumonia, a particular bacteria which causes pneumonia disease in living organism. Okay, जो bacteria pneumonia disease cause करते हैं. So he took one living mice and a bacteria that cause pneumonia disease. The name of the bacteria is Streptococcus pneumonia. Okay, all right. So what he did before that, the Streptococcus pneumonia, he showed that the Streptococcus pneumonia produces 2 types of body. Okay, 2 types of body. One is with smooth body, the other one is with rough body. Okay, the Streptococcus pneumonia, when they reproduce, they produces 2 types of body. One is with the smooth body and one with the rough body. The smooth body is due to capsulated body, the capsulated layer that is present outside the body. Their body wall is covered with capsule, okay, and this capsule produces thick mucous substances. Okay, thick mucous substances. सब sentence में नहीं लिखूंगा, ठीक है? So this, why it makes it smooth? Because their body is covered with capsule which provide sticky, slimy mucous substances. While in case of rough, it is without capsulated. That's why it makes a rough body. So non-capsulated. So their body is not capsulated. That's why it makes them a rough body. Okay, so that is the differences. One more thing, the smooth Streptococcus pneumonia, they are the virulent. That means they causes disease. They are pathogenic. They are pathogenic or they are virulent. They are the actual Streptococcus pneumonia which causes disease. Rough Streptococcus pneumonia are non-virulent. They are non-pathogenic. Okay, so scientist Griffith pathogenic. He found out that the bacteria produces 2 types of body, one with the smooth and one with the rough. The smooth one is the actual bacteria that causes diseases, Streptococcus pneumonia, and the rough one, they do not cause any diseases. Okay, so they are non-pathogenic. So what they did, scientists extracted separately these two bacteria. Okay, the Griffith, they extracted separately smooth one and the rough one, and they put them in a petri dish. Petri dish means it's a plate like substance. Plate जैसा होते हैं structure. Okay, plate is a plate like substance. Okay, जहाँ पे when researchers or scientists, they do their research on bacteria, they always do in a test tube or in a petri dish. Petri dish, okay, is a kind of plate like substances where bacteria are kept along with their media. So Griffith extracted various smooth Streptococcus pneumonia and put them in a petri dish. So he gave them a name, strains, strain 1, strain 2, strain 3 and so on. ठीक है, बहुत सारा एक्सट्रैक्ट किया गया स्मूथ स्ट्रेप्टोकोकस निमोनिया को, इसके बाद उसको अलग अलग प्लेट में रखा गया. ठीक है, then he gave them name strain 1, strain 2, strain 3, strain 4, 5, 6, 7, so on. Similarly, he done for rough also. So he gave them a name R1, R2, R3. Okay, strain R1, strain R2, strain R3 and so on. Okay, whenever we put some bacteria in a petri dish or in a test tube, we give them a name. If we give them a name, and if we have suppose 10 test tubes, so we give a name strain 1, strain 2, strain 3, strain 4 according to our concern. So smooth one, he gave them a name S1, S2, S3. He took out many smooth bacteria, he reproduced many smooth bacteria and put it in a separate plate S1, S2, S3, S4. He gave them a name. Okay. Similarly for rough one also, he took out various rough bacteria, put them in a separate plate, petri dish plate. Okay, many petri dish plate. So he gave them a name R1, R2, R3 and so on. Okay, বুজি পাইছা? So দুটা টাইপৰ body form কৰে Streptococcus pneumonia. এটা হল rough body, এটা হল smooth body. Rough কিয় কাৰণে হয়? Rougher body layers না থাকে. এই layers তো capsule বুলি কওঁ, layers পোৱা না যায়, হেতু কাৰণে body rough হৈ থাকে. So হে rough bacteria খিনি actually কি হয়, non pathogenic disease cause নকৰে. Smoothত কিয় smooth কোৱা হয়? তাৰ body layerত capsule পোৱা যায়, হেতু capsule কি কৰে, এটা thick mucus বিজলি বস্তু এটো produce কৰে আৰু. So হে কাৰণে তাৰ body smooth পোৱা যায়. So egeta ki hoy pathogenic hoy, they causes disease. Okay, so this we have. So for his experiment, Griffith took the smooth S3 and the rough R2. This is important from entrance point of view. So he chose S3 strain. When we talk about S3, that means smooth one, and R2 strain. Strain बोलते हैं हम लोग उसको. Whenever we keep any microorganism in a plate or in a test tube, we call it as a strains. So he chose S3 and R2. Maybe he found out that this is the correct one, or it is the best one to do his experiment. So he chose these two. So now he performed his experiment. What he did, so R smooth means pathogenic, rough means non-pathogenic, right? We have known so far. So experiment, what he did, the first one, he injected S3 strain into a mice, living mice. When we talk about this mice, living mice. So ki korile S3 strain mice or body inject korile. Okay, S3 means was smooth Streptococcus pneumonia, right? And it is virulent, it causes disease. Then he found out that the mice died due to pneumonia. Okay, because of course it is a virulent, S3 strain is a virulent pathogenic. So he found out that, yeah, we can also understand that it is a virulent. Definitely it will lead some diseases, it will cause some diseases into the mice body. So mice died. Next experiment, he injected RAF2. Now the reason why he chose S3 and R2, maybe he found out that these two strains are the best one to perform his experiment. So that's why he chose. Okay. The remembering part is which strain he chose. You need to remember from entrance point of view. So RAF2 is non-virulent. That is the RAF1. He injected into the mice. So it is a non-pathogenic, non-virulent. So ultimately we can come to our conclusion that the mice will definitely live, right? So he found out that mice live. So mice survive because it is a non-virulent. So this is the simple thing we can also do, right? That one is 2. So next, what he did? He hit kill S3. Okay, he hit kill S3. Hit kill means he burned it for some time. Okay, he put in a test tube the S3 strain and he hit it for some time. So next, what happened? What he did? He injected hit kill S3 strain. Hit kill means he put S3 strain, that means a virulent smooth bacteria, into a test tube. Hit kill means he hit it on a fire for some time. Okay, that's why we say hit kill, and injected into the mice. Injected into the mice. Now what result he found? He found out that the mice live, the mice survive. When there is a hit kill, he found out that the mice live. Okay, S3 strain is a virulent. When injected into a mice died. But when we heated this S3 strain, he found out that the mice live. Okay, he found out that the mice live. Now next, what he did? He combined two things. Hit kill S3 strain plus R2 strain. He combined those two things, hit kill S3 strain and RAF2 strain, and injected into the mice. Now what result we can observe from here? What should be the result we can observe here? When heat was killed, when S3 was killed by heating, the mice live, and the R2 is the non-virulent. Okay, R2 is the non-virulent, they do not cause any diseases. So we can, think that mice would have lived. Okay, mice would survive. Because this one is also heat kill and this one is also non-virulent, mice would survive. This we can think. But he found out that the mice actually died. Okay. Now what result we are supposed to get here? Because it is a heat kill, mice live, mice should have lived, and it is the R2 non-virulent, mice should have lived. This is the result we expect, but he found out that the mice died. So from this experiment, he came to the conclusion that the S3 strain must have transformed something into R2 strain. Okay, strain means there is a cell in it. Okay, cell, the cell of the smooth and the rough bacteria. So from this experiment, he came to the conclusion that the heat kill S3 strain, though it was heat kill, there must be something still present, which something transformed into the R2 strain, which was non-virulent, now resulting into the formation of virulent. Okay, So heat kill S3 strain must have transformed something into the R2 strain and transform R2 strain into pathogenic. Okay, so he just came to the conclusion that something has been transferred to the R2 strain and result in the transformation of R2 into pathogenic. So that's why mice died. Okay, so this was the first and initial experiment done by Griffith based on transformation to prove that DNA is a genetic material. But this experiment do not give us a conclusion, exact conclusion that whether it is DNA or any other molecule which has been transferred. Okay, but this is the initial experiment, so it has a very important in genetic material of DNA. So from this experiment, other scientists again perform another experiment. Based on this experiment only, they just added some additional things and perform this experiment, similar experiment. Okay, so far he came to the conclusion that when the heat kill S3 and R2 was combined, the mice died. So from that he found out that, or he just examined or analyzed that the heat kill S3 strains must have transferred something into the R2 strain. R2 strain is non-virulent, they do not cause diseases, but when he transfers something into the R2 must have transformed into virulent. That's why mice have died. Okay, so but he did not give exact conclusion whether it is DNA or any molecule has been transferred, but he just gave us some idea that something has been transferred. Okay, so based on this experiment, other scientists perform similar experiment but with certain additional things. We will see this in the next video lecture.